start now. Fine. And I will share screen on this slide on my notes. Okay, so I will make a small jump, which is to a new topic. So I have two slides about, two lectures about this topic. And this topic is a different one. It's a new one for me. It's the first time I explain, so could you give me the feedback to see if you understand it or not, okay? So I'll discuss in the next two lectures, the last ones of the course, the problem of chemoreception, which is about biological systems sensing chemical cues, okay? So one example, mainly I will focus on this paper, Berg and Purcell, Biophysical Journal, 1977, okay? This is actually a classic paper. It's one of the most famous papers in biophysics, okay? So this paper discusses, is a theoretical framework to discuss how a biological system can sense its environment. And sensing is important in many situations. Okay, here you have your friend, the run and tumble process, in which a bacteria can change its way of moving. For example, tumble means the exploration, like doing diffusion all around, and then it can switch to a new mode of moving, which is running, okay? And tumble again, tumble for some time, and then run, okay? This type of motion is useful, for instance, to find a target. So for example, when the bacteria are hungry and they want to find food, you can use this type of motion to find optimal strategies to find this food. For example, to finding what is called an attractant, a chemoattractant with food, okay? So in here, it is important two things. First, to sense how far you are from the target, and then second, to find it. So you need to, the bacteria has to sense, for example, here there is lower concentration than here, and then it has to move in this direction. So first is the problem of sensing, second is the problem of decision making, third is the problem of how do you move, okay? The same thing, it happens in cells. So imagine you have a tissue like the one I'm drawing here. Okay, this is a sketch. The tissue is made of cells, and this tissue needs to grow. For example, if you cut, you make a cut with a knife, you open a wound, and then you need to recruit cells and to make that cells proliferate and they grow. So this is a problem of decisions, cell-fate decisions, and it's a problem in which the cell has to sense that something is wrong, and it has to take and to start proliferating. To do this, the cell has to sense its environment and understand that something is going wrong, and understand what is happening with the chemical signals around the cell in such a way that this triggers some signals inside the cell, and this produces that this cell, for example, divides into two new cells, okay? So in essence, this is a very important problem. So as I said, here are some examples. One is the one I was discussing before, which is chemotaxis, which is a motion driven by chemical signals towards or away of food or toxins, for example. So it could be there's a toxin and you want to escape from it, okay? So it's not only chemoattractants, it's also chemorepellents, okay? Another one is very important is cell-fate decisions to cellular signals. So for example, I also didn't talk about stem cells. So you know, stem cell is a cell that can become different things. It can become a muscle cell, it can become a epithelial cell, and so on. But this is cells that also sense the environment, and depending on what is happening in the surroundings, they take one decision or another. As I said also, well, development, differentiation. This is also important. Development when the embryo is growing. So the embryo has, when you have an embryo, the cells have to know or to realize that they can grow, but they can grow up to a limit. You cannot have an infinite growth in these problems. And another one that it is important in this type of sensing is the photoreception in the retina. So you should know that in the retina, in the eye, we have cells that can sense single photons. So they can they are sensitive to the arrival of a single photon. This is very, very spectacular. And in my last lecture, I will discuss a model for this. And in the first lecture, I will give something more about general model of sensing. Okay, so this is just a motivation of what will come later. But just to let you know, if you are interested, there are a lot of topics about this. Okay, so I stop here. And okay, now I lost my. I'm sorry, but I lost my slide. So give me a second, because I'm not totally lost. I don't find it. Okay. You see my whiteboard now. Please let me know, because if not, I don't know. You see my whiteboard or my notes?
Yes,
the whiteboard. Okay, very good. So, okay, so this is lecture. This is very thick. Lecture, I don't remember now. 23?
24.
24, which will be about sensing the environment by cells. 1. And as I told you, I will discuss, okay, this is very thick, I don't know why. Fine. I will discuss mainly a paper you can read very well right now, which is the following: Berg, Purcell, Biophysical Journal, in 1977. Okay, so all I'm going to discuss today is in this paper. Well, some parts at least. In this paper, they discuss the physical limitations of a cell. It's a model of a cell, so that's why I put it like this, okay? Of a cell to sense and respond, to changes in its environment. Okay, so physical limitations, by this I mean that fluctuations will be very important. So the idea that I'm thinking about is, for example, you have a bacterial cell, but it could be also a human cell that is swimming. And this cell has, of course, receptors in the membrane. And these receptors allow this cell to sense how many molecules are in its surrounding environment. So we consider there is an environment, there's a volume around the cell in which there are molecules that can be sensed by or counted by this cell. Okay, and I will give 2 models in which this cell can count the molecules in the environment. The first one is okay, very simple. It is called the perfect instrument in this paper. Perfect instrument that makes a single count of how many molecules are in its environment. Okay, it makes a single count. So it's like you have a camera and you make a photograph, and in this photograph you can count all the molecules that appear at a given time in this photograph. Okay, so it counts all X molecules. X means not the position. X doesn't mean how many molecules. X means that these molecules are of a species called X. X could be, for example, I don't know, CO2, for example, no? Or X could be, I don't know, an enzyme, or X could be whatever. It's the species, okay? Counts all X molecules in volume B. Okay? And we will say that the object of, the purpose of this cell is to estimate the concentration. So we will say that the real concentration is C bar. Okay. And now the first thing we would like to know, okay, this cell makes a single shot, a photograph, and it counts n. N is the number of molecules in this volume. Here will be 1, 2, 3, 4, 5, 6, 7, 8, for example. Okay, this is an example. Of course, if you take a photograph, or you take a photograph later, or in another moment, there will be fluctuations. So in reality, this will be a fluctuating number. It depends when do you take how many particles were passing by here, by this volume, when you take the photograph. So what is important is the expected value. How many molecules we expect to count. The expected value of n is just the volume times the expected concentration. Okay, this is what we expect in a single snapshot. This is a number. So imagine I tell you, I prepared an experiment and I have 1 millimolar. So if I take a snapshot in a volume, I expect to measure this. Okay, this will be my average expected value. And an important point is what are the fluctuations of the number of molecules. Of course, the molecules are here, but they can also exit this volume. So there are fluctuations in this volume. So in a very simple estimate, you can say that in a single count, the fractional error in a single count, we will define it like this: delta C divided by C bar. C bar is the expected value. So this is kind of the mean. And this is the standard deviation. Okay. Standard deviation over the mean, what you expect, if you do very simple theory of counting, would be something like this: 1 over the square root of the average of n. Okay. This probably you got in probability theory courses. You would expect the relative error is of the order of 1 over the square root of n. Okay. If you have many molecules, you expect the fluctuations are small. So this goes like 1 over the square root of n. Okay. So what does this mean? This means if you do this approximation, something like this means that this goes like volume times C. So I'm just replacing this equation here to the power minus 1/2. Okay. This is my fractional error, what I expect to have as an error in the measure. Okay, so this will be in a single shot. So in a single shot, I make a photograph. I expect to get a fractional error, a relative error of volume times the concentration to the power minus 1/2. So now, what it makes sense is, what if we repeat measurements? So instead of making one photograph, we make a series of them. So we have the camera open for some time. This is very important. So we would say, I would like to repeat measurements to reduce the uncertainty. Okay, this is the uncertainty we have in our measurement. So what is important is that, let's say we are given a time window, 0t. They tell you, okay, I give you 1 second, and I let you do in 1 second a certain amount of photographs. So how many of them are meaningful to do? So it is important to say that I would like that the photographs are independent. So there is no correlation between the photographs. That's a way that I'm taking independent samples. So how many independent samples we can do? So this would be how many independent photographs. It would be the total time divided by the escape time of a molecule. Okay, so if I let the molecules escape, this will be like reshuffling everything again and taking an independent photograph. And the main point here is, what is this? The escape time of one molecule. And this, if you go back one molecule, sorry, I was thinking in Spanish. If you go back in my lectures, you can find that the escape time in a volume, you take one molecule, how long it takes to escape a sphere, this goes like L squared divided by D. Okay, but L would be like the ratio, the radius, okay? Well, actually, the best is I call this A, because later I will use A, okay? So this will be A squared divided by D. And this is the diffusion coefficient of the molecules. A squared, in reality, goes like the volume to the, okay, A is volume to the 1/3, because the volume goes like 4/3 of pi times radius to the cube. So this is like B 2/3 divided by D, okay? Remember, here I'm using that the volume goes like A to the cube, okay? It's 4/3 pi cube. This is what I use here, okay? So the time to escape for a molecule is like the volume to the power 2/3 divided by the diffusion coefficient. Okay, now, how many, sorry, you have a question?
Yes, Professor. Why we count the number of molecule?
Because, okay, this comes from the beginning of the lecture. So imagine you have the bacteria wants to find where is a source of food, okay? This source of food, it will be like, okay, you have the food here, and this is like, this food is diffusing in the environment, okay? So of course, there are a lot of molecules here. This is highly concentrated, but then the molecules diffuse around. So there is a concentration gradient. So if you look, this is, for example, dimension X. Here, there are a lot of molecules, and this is decreasing like this, okay? You follow me with this?
Yes.
This is C of X, concentration of food molecules as a function of the distance to the source of food, okay? So imagine the bacteria is here. The bacteria, or the cell, it's hungry. So it wants to eat, it wants to find this place, okay? In order to find it, it has to decide how to move. So if suddenly this bacteria sees there are 3 molecules here, but 2 molecules here, the bacteria will say, okay, probably the food source is going to the right, right?
Okay.
So it is important to count, because this way you navigate efficiently. You say, okay, I don't know where is the source of food, but I've seen that here there are more molecules than here. So it's like if you are walking blind, but you get some impacts of molecules, and you want to know what is the source of the wind, for example. Then you will do like this. You will count. You will feel. Well, in one case it's the velocity, but for this cell, you will count. This will be your way of orienting yourself in the environment in such a way that you find this target fast, right?
Okay. That means the number of molecule impact on the run and tumble motion.
Sorry?
Right. I said that means the number of molecule we have it have the impact on the run and tumble motion.
Exactly. So if the bacteria is clever, suddenly it finds a lot of molecules close by. You say, okay, now I run and I go fast.
Okay, thank you.
However, if you are in a position where they are the same behind than in the head than in the tail, then the bacteria will say, okay, I will be tumbling, I will be searching around. Okay?
Okay, it's clear.
Fine.
Thank you.
Other questions? No? Okay, fine. So now what I'm discussing is how many independent measurements of photographs I can do in time t. So this is the total time divided by the escape time. So it's the time I need to reshuffle my statistics. This is just t divided by this. If I divide by this, it's td divided by p to the 2/3. This is the number of independent measurements in 0T. Okay. So this means that I can reduce my relative uncertainty because now the number of measurements that I will make with this scheme, repeating measurements, will be larger. I can do more. So instead of n, I will have n times this, okay? So the number is increasing. So now this implies that delta C over C in this scenario is 1 divided by this number, which is, okay, I have a square root, don't forget, TD divided by B 2/3 times the previous number that they had, which is BC square root. So if I multiply all this, what I get is, okay, the T goes up, T, D, and C bar. I put this with -1/2. And then V, I have B divided by B 2/3, which is B 1/3 with 1/2. So it's B to the -1/6. Okay, so I get this formula if I can take more than one photograph in time t. Okay, more than one measurement in time t. Okay, I copy this equation just for convenience. Delta C over C bar equals TD C bar -1/2 times V -1/6, right? So now let me assume that the volume is a sphere, so it's spherical volume around the cell. The volume will be 4/3 pi A3. Okay, we have a sphere. Okay, so like this of radius A around the bacteria, for example. Okay, this is the region where the bacteria can count molecules in our model, right? So now here we replace this volume, and what we get is delta C divided by C bar. It will be 4/3 of pi to the -1/6, pi to the -1/6 times TDC to the -1/2, and then there will be also A cubed to the -1/6, which is A to the -1/2. So you get TDC bar A to the -1/2. Okay, so what you learn from here is that this is a number. So 4/3 of pi to the -1/6 is a number. You can write it. So actually all this number here is 1.6. You can write it like this, 1.6 to the cube. So it's T C bar A to the -1/2. Okay, this is the formula that we get with this simple estimate. And now let's put this formula in practice and see what does it mean. Let's give an example. Okay, first of all, from these formulas you have seen the first important message is that the relative uncertainty goes like 1 over square root of C. Okay, this is a very important result. It's, I think, okay. This is a classic result in biophysics. It has been also tested in many experiments. So this is, just to let you know, this is in gray, but it seems I cannot do much about it. Okay, fine. This is a classic result in biophysics. It is important. It has been tested in experiments as well. Okay, that cells, experiments. Cells can sense the environment, and the error that they make in measuring the concentration is of the order of 1 over square root of C. This is a very important result. And it can be proved in different ways. You will see it will come also in a more complicated one. Next is, let's try to use this to give numbers. So one example, let's see how this works. Okay, example. One example is the following. We have a cell that has a can measure around a volume of radius 1 micron. Okay, this is reasonable. The second is what is the diffusion coefficient? This D. The diffusion coefficient we can take the same as for example, for one ion. Let's think a cell counting ions. This we took like 10 to the 3 micron cubes second. Okay. Again, this is an example. Let's say I would like to know, for example, how long. Okay, the question will be the following: how long I need to measure in order to determine this concentration of, for example, 1 micromolar with an accuracy of 99% accuracy. Okay? And the question is, how long does the cell need to measure? It means you have a device and you put this device to count molecules. How long do I need to count molecules in order to measure this concentration, which is very small, 1 micromolar, with a 99% of accuracy? This formula gives you an estimate for this problem. How long? So how long means I would like to know T. Okay. Again, T is this one, is in micron cubes per second, A is in microns, and C is in micromolar. But I show you in my previous lectures, I show you how to convert this into microns. Because, okay, I open a parenthesis, this is micro is -6 moles per liter. Molar is in liter. Then we knew that 1 femtoliter, 10 to the -15 liters, is 1 micron cube. Okay, and then we also know that 1 mole is 6 times 10 to the 23. So this, if I multiply by all this, what I get, so this, this and this is in units of micron minus cube. So I will get the concentration in micron minus cube, A is in microns, D is in microns cube. Sorry, this is a mistake. Diffusion coefficient is length squared over time. So micron squared divided by second. So all this will have the right units because I will get 1 over second and T is in units of second. So this will make sense. So if I multiply all this, I get 600 microns minus cube. Okay. This C bar is this. Right? So I want to know T. So how do I do T? So in reality, it's a delta C divided by C bar. In reality, is 1 over square root of 1.6 T D C bar A. This is the formula. So I want to know T. I put T on the left and I square. So T is equal to this one in the numerator, C bar over delta C squared divided by 1.6 times D C bar A. Okay? All right. So here I have everything because I know D, I know C bar in micron minus cube, I know A. This gives me, all this part gives me a number in units of 1 over seconds. It will go to the numerator, so it will be seconds. And now I need to put this number. So what do I put for this number? And this, I open the question to all of you. What is C bar divided by delta C? But actually, it's easier probably if we write this one. What is delta C divided by C bar? Can anyone guess or say how much is it? Nobody?
1 over square root of 600.
Square root of how much?
600.
600.
Okay.
1 over square root of C bar. So C bar is 600 micro.
Okay. be careful with this, because what it's appearing here in the formula, this is the concentration you would like to measure. What is appearing here is the accuracy. I could call this epsilon c, okay? This is the accuracy you want to measure, and this is the real concentration. Be careful, because here I'm saying I want this accuracy, okay? This accuracy means, okay, so if I am 99% of accuracy, it means that I have an error of 1%, no? Right?
Yes.
Okay, an error of 1% means, in fraction of 1, means that my standard deviation divided by my mean should be 0.01.
01. Ah, okay.
This is what it means, okay?
Okay.
This is the way you have to understand this formula. You can also write it like this, epsilon c equals to this, okay? This is the accuracy, the left. This is the accuracy. Well, not accuracy. In reality, is accuracy would be the opposite, C bar over delta C. It is the uncertainty in the measurement. Is the relative uncertainty. And this, the C that appears here, is the value of the concentration you want to measure. Value of the concentration. That is the mean value. It's also the mean value, okay? But here is mean, sorry, is standard deviation divided by the mean, okay? So of course, you can use this to get the standard deviation. Use these two together, can give you the standard deviation, fine, you are right. But if they tell you, okay, I want this accuracy, you can just say this one that I'm showing here, okay? So in other words, just to simplify a bit, not very efficient, not too clean, but okay. I just want to say that here we can just, in this place, put, this is, remember, delta C over C bar is 0.01. So C bar over delta C is 100. This is 100 to the power 2. And in the denominator we have 1.6 times D, which is 10 to the 3 micron squared by seconds, times times C bar, which is 600 microns minus cube, times A, which is 1 micron. If I do this, micron, 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 they go. There is the second who goes up, and what you get, T equals 0.01 seconds. Okay, which you can also write it in milliseconds. So this is 10 milliseconds. Okay, so a cell in this rough estimate, it's not that rough, we did some theory. A cell who wants to measure with 99% of accuracy this concentration using this radius and molecules of this diffusion coefficient needs to measure but for 10 milliseconds. Okay, so you see, this has some practical application because it's, well, in reality, if you have a device that is measuring, counting molecules, the theory will be the same. Okay, this is fine. And now, okay, this is example A. If I do example B, it's another example in which I tell you how long I have to measure to get 90% of accuracy. What do you think? What do you think will be T? Will be bigger or will be smaller? Without looking at the formula.
Is it smaller?
It's smaller. Any guess why? So if you want to be more accurate about a phenomenon in physics or in biology, if you want to be more accurate, you will need more information, no? About a physical process, right? So you will need more time. you need to measure more in a system if you want to be more accurate. Okay, if I go this way, I will need to measure in longer time. So if I go in this way, if I don't care and I don't want to be so precise, I can take less information and go faster, no? So I will need less time to do a less accurate measurement, right? Is this the logic you were thinking about? No. You have doubts?
Yes.
Yes.
Okay. That's the idea. So you have a device, you have few counts. This will be maybe 90% accurate. But if you want to be more accurate, take more and more and more information. So if you do this, 90% accurate, what you get, you repeat everything. Okay, what you have to do is that delta C over C now is 0.1. This is 10% of error in reality, okay? So if you put this here and you repeat everything, you get that T equals 0.1 millisecond, okay? So we have changed here the accuracy by a 9%, and a 9% of accuracy implies that I can reduce the time 100 times, okay? So if I make measurements in time windows that are 100 times bigger, I increase the accuracy of 9%, okay? So it's not bad, this one. So you can do very short measurements and still be very precise, 90%. Okay, this is the idea. All right, so if there's any doubt of question, okay, so the conclusion of this is that increasing the accuracy has a time cost, okay? As we expected. You need to take more information in order to be more precise, right? So this was the first model. There's a second model, which is, I think, more beautiful, but also a bit more complicated to solve, which is discussed in the paper, which is, they call it the perfect instrument.
So
Yes, please.
if we increase the time, in some experiment, when we increase the time, at some point we remain at the same place. It's like the same accuracy. And increase the time will not increase the accuracy. It's not the case in this case.
No, okay. I don't understand your example.
It's like we are doing an experiment, and we took something like 5 minutes to do the experiment, and we get an accuracy. We repeat the experiment again and add the time like 10 minutes, but we still get the same accuracy.
No. In reality, here the accuracy is changing because if you observe the system for a longer time, you take more photographs, okay? Yeah. And if you take more photographs, you have a bigger statistical ensemble, and then you reduce the uncertainty on it. Okay, so it's like you have a phenomenon that has fluctuations, and the more you look at it, the more you will know about it, and the less uncertainty you will have about it. That's the idea.
I get it, but we don't have some time where the number of particles started repeated. Like we get like 5 before we get again 5.
Yeah. If you do a model where there are few particles, this will happen. But the approximation I'm considering is that there are many particles. Okay.
Yeah.
This will come now when I do the math. But I'm assuming there are many particles. Many doesn't mean 3 or 4. It means thousands.
Yeah, it's just the example I took.
Yes. The example you took, it goes in a limit in which biological systems don't operate, in which you have 2 or 3 molecules. This is very rare, okay? I'm discussing a system which has, okay, if you can look at it, because here there are 600 molecules in a micron cube. So the volume that we have is of the order of a micron cube or more. So there are, on average, 600 molecules in this system, okay? So I think the situation you are thinking about is not happening in this limits. Okay?
It's like, okay, we have thousands of particles.
Yes.
And in the first measurement, we observe like 5 million.
No, this won't happen. This will be very difficult. Okay, so let's say you have, okay, you have a box, and you expect to have 600.
Yeah.
The normal fluctuation will be, I don't know, you have 540, for example, makes sense, or 525, or 631. These are okay, these numbers, because this lives in a bigger volume. So there are more molecules around, so they can enter in and out, okay? So these type of fluctuations are expected. What I'm saying is, if you want to measure the average accurately, it's better you take many snapshots than if you take one.
Yeah.
Okay, so if you take one, you could get this one, but you could also get this one, but you could also get this one. But if you integrate and sum measurements, so if you, instead of taking only one, you take 10, and then you take 10, you get 10, and you average, this has less uncertainty than taking only one. This is what I'm saying.
Okay.
Okay. Think about it, and if you have more doubts, we discuss more in detail. Okay.
Okay.
Okay. Okay, very good. So this was the first model. The second model I will discuss is a perfect instrument with time integration. Okay. Time integration will be something similar to what I just talked about. What I was telling was like, I take different snapshots and I accumulate that information. Now, you will see this, give me a second, I need some light. You will see this in a different way. So we will think about the following model. So we are thinking like the cell is like an antenna, no? So the cell is like, we have something like this, okay, I should copy my drawing because then I don't, imagine you have an antenna, okay, and then this is something like this, and this antenna can count all that is around in a volume. The volume that I'm thinking about is something like this, okay, so it's a sphere near the antenna, and this sphere has okay, this shape. This has radius A, and this antenna can count all the molecules that are around in this volume. Okay, well, imagine if there are, this is a very crude example, 5, this antenna can count 5. But now we say that the cell is a counter that counts the exact number empty of molecules within a sphere of radius A, which I will call it SA sometimes, okay? At time t, okay? So MT here is the number of molecules at time t in a sphere. And we will say that the way this device works is that it does time integration between a time ti and a time ti plus t, okay? Before I was saying 0 to t, but now, okay, time 0 doesn't exist for a cell. So it will start measuring at ti, this is the initial time, and then we'll measure for a time window of length t. All right, so as I said, the way we will estimate C bar, we will have an estimate. C bar is the real value. Our estimate will be called C. And this will be basically the number of molecules. So it's a concentration. Concentration is number of molecules per unit volume. So we will do m of t. This is, okay, I'm using different notations. So let me call this, I think this is easier for later, because I have two notations. Okay, no, sorry, this is correct. This is correct, T. Okay. You will see something very important, that is, what is m parenthesis t is the number of molecules at time t, and mt is the integrated, the number of molecules counted in this time window. This will give me the m of t, which I will define below, okay? So the estimate will be m of t divided by the volume, number of molecules divided by the volume, and the volume is 4/3 of pi a cube. So you get 3/4 pi a cube times mt. This will be my estimate of the concentration from this interval. So what I'm doing, okay, and this is important, is that I am measuring in this interval. And I can measure at all times in the interval. This is what it's called here, time integration, okay? So if I call time integration, means that the following, that what I can do, this cell can do the following, mt, which is a time integration of the instantaneous value. So what it does is it sums the molecules at all times between time ti and time ti plus t. And then it averages this number by the total time, okay? So this means I have the device measures at time ti, then later it measures again, so it has a window where it's all the time measuring. It sums all the number of molecules in this window, and it divides by the time duration of the window. So this is like the average value of m in the window, okay? So this is the average number of molecules in the volume in SA in the interval ti plus t, okay, by doing time integration. Okay, so now we are saying we have a device that can do this. And what I'd like to know, so this is a fluctuating quantity, because if I have a time window, I can count in this time window, I don't know, 1,000 molecules, but in another time window, 990. So this has fluctuations. I would like to know what are the fluctuations, particularly this. What is the variance of this? Okay, what is this? Okay, these are the fluctuations. So first, in order to know this, I should know what is MT, average, but this is simple. MT average, it's simple because we know what is C bar. So C bar is the real concentration. And if this is a good measurement, C bar should be the average of this. So it should be MT, well, this one, 4 pi a cube times MT. Okay, this means that I can solve directly from here and get that MT equals 4 pi thirds. Okay, sorry, I had a chat somewhere. Done it. Okay, now they say they stop recording, but I need to record now. Please keep the recording. Sorry. Okay. The recording is okay. The recording is continuing. Fine. This is very complicated. Okay, so I close the chat. The average of MT is related to the real C like this. So 4 pi thirds a cube sigma. Okay, this is the first part we need to solve. to know the fluctuations, the average. Okay, I know the average, but now I'd like to know MT squared. And this is the most challenging part of this model, which is what is MT squared, the second moment. Okay, so I'll do a short break, but first I'll write the formula, and then I do a short break and continue the calculation. So MT squared without the average is the square of this. And be careful, because this is related to your Brownian bridge problem. So when you have this and another thing that looks the same and you want to do the average, be careful, because there are two time indices. So the way you do this is as follows. Some of you said, okay, I take this MS, I multiply by the other square. The correct way of doing this one, so multiplying this times the same, is you put in the same here, you put MS prime, okay? And then you see what happens. This is the right way of doing, because you can mix different types, okay? This is the way. Be very careful with this. I've seen some people who make mistakes in this thing, which is in principle simple. So the way we do this is the following: 1 over t squared. Okay, this is obvious, this is a prefactor. And then you have these integrals, ti plus t ds prime integral ds ti plus t, and then you have ms prime. This is the way we have to, okay? These are dummy variables. You can call them as you want, s and omega, or s and z, but be careful with this. This is something I saw in your exercises, okay? So now, okay, let me continue a little bit, and I do a short break. The average of MT squared is, okay, 1 over t squared, integral 0 t. Okay, here I used steady state, because it doesn't matter if I put ti plus t, then I put 0. We are in a stationary state, okay? We assume stationary state. Stationarity. So I will take from 0 to t, which is simpler, and it will be ds prime integral 0 t ds of ms prime average. So the only place where I do the average is here. Okay, now there's also another chat. I don't know what is happening. Okay. Some part, okay. Kirel, I don't know if this is in. You send a message to everyone. If you have a question, please. I don't know if this is English. This is probably in Filipino, no? Okay, no problem. This is 1 over t squared. I will just do a definition here. ds prime integral 0 t ds, and I will call this object, g of s minus s prime. It's like a correlation function. G s minus s prime. Okay, and then I have introduced here the following. G, I call it tau, is defined as mt plus tau. Okay, this is usually called, as I told you, the autocorrelation function. Remember, we are in steady state, so this obeys g of tau equals of g minus tau. Okay, I will do a short break here and see how far I can go. So I stop sharing. No, first, stop share, and then I'll pause the recording, resume recording. All right. All right, so I define this quantity, which is important for this calculation. I call it autocorrelation function. It's just this average, remember. And let's see if I can. Okay, it's too small. 1 second. So what I'll do now is try to calculate this quantity. So to do this, I will try to show you another definition of empty. So what is empty? Empty, in reality, is, so we are counting at time t how many molecules are in the volume A. In reality, it can be written as a sum from j equals to 1 to n of Wjt. What is n, what is w, what is everything? Okay, so what we are saying is we have the sphere. This is the sphere of radius A. This is SA, radius A. And we have a bigger sphere nearby that is following, is surrounding this sphere, and it has a bigger radius, which is big R. So we are saying R is much bigger than A. This is an assumption. Another assumption is in the volume R, so this would be like, for example, a full container where the cells are moving, there are a total number of n molecules, molecules in the big sphere SR, okay? And this is conserved. There are no creation or destruction of molecules, okay? There's a total number of n molecules, and a fraction of them are inside the small sphere, okay? So this n is the sum over all the molecules, also including those that could be out of our sphere, okay? So what is Wjt is the following. Wjt is a counter variable. So it takes the value 1 if the molecule J at time t is in the sphere. So if the molecule, let's say this is a molecule J, is out of the sphere, this W will be 0. And if the molecule is inside the sphere, the W will be 1. In this way, we will count only those that are inside the sphere of radius A, okay? This is if J of t is not in the sphere, okay? These are counter molecules. This is a way we define this M. So now we can put this M inside this definition. So G of tau will be just, well, we'll have the first sum, J equal 1 to n of Wjt, multiplied by the sum I equal 1 to n Wi t plus tau, okay? Remember, when you do this type of products, it's better you put the indices with different letters, okay? This is very important. You will see now why. It's very important because now, let's say if I do different colors, this is one molecule, and this is another molecule, the yellow one. It is very different if you consider two of them that are independent. You do W, for example, W1 times W3. These are two independent molecules that if you have W3 times W3, this sum contains all, this sum contains all. This sum contains the red and the yellow, and this one also, the red and the yellow. This is a time t, this is a time t plus tau. So you have to be careful and separate these sums. If you continue, you see that there is one term that is mixing cross terms. So the molecule 1 with all the rest of the world, the molecule 2 with all the rest of the world except itself, and so on. So this first term will be this part, the sum, double sum, okay, and multiplying, I equal 1 to n of Wi t plus tau, then it will be Wjt, and I will sum this molecule with all the rest that are not itself. So J distinct to I up to n. Okay, this is the first term. The second term will be the first with the first, the second with the second, the third with the third. So this will be just the sum I equal 1 to n. So you're summing over the same of Wit with Wit plus tau. Okay? This is like self-interaction, and these are cross-interactions. Okay? Do you follow me with this?
Yes.
Okay, very good. So this one, I will call it G1 of tau. And this one, I will call it G2 of tau. Okay, I will first calculate the cross terms and then the self-interaction term. So let me go with it. And the first term, G1 of tau, remember it was this, the average of J different to I, Wj t, Wi t plus tau. There is also sum over I inside here. I equals 1 to n. So these two are independent. So there is independence. Because of this, independence of I different to j. Because of this, we can break this average and write it like this. Sum j distinct to I to up to n, sum I equals 1 to n of omega j. So Wjt times Wi t plus tau. Okay, we can write it like this. They are independent. The average of a product is the product of the averages. Okay, so now something very important is, so here all the terms are the same because the average of all of them is the same. So this one is equal to this one. And there is also steady state. So it doesn't matter at which time you look, it is the same average. So this will make that this sum will have a lot of terms that are the same. And how many terms is, okay, you have n sum, and here you are n minus 1. So this is n minus 1 times n times wi, sorry, this is not very good, times wi squared. Okay. Sorry, not wi squared, it's wi to the power 2. Okay, because it's the same one. Wi times wi squared. Here we use stationarity. Stationary. And all the particles are identical, and identical particles. Identical particles. Okay, so we have this, and now we know from before what is this one. This wi square is the average to square. We calculated the average square. So wi average, we have it from, actually it's something very similar to this, but I didn't write it. So I forgot what is this to average, okay? So what is the average of Wj? It's very simple. Okay, I think I don't know, I don't need this red. It's not that important, but it's important for the calculation I'm doing now. Wj average, this is important, is, okay, I have big N molecules in all the volume, and some of them are in the inside volume. So the probability that I am inside, so that a single molecule is inside, is just the ratio between the volumes. It is just the volume of the small sphere divided by the volume of the big sphere. So it's 4/3 pi a cube divided by 4/3 pi r cube. This goes away and it's just a cube divided by r cube. This is the average of Wj. Okay, it makes sense to you, no? So I have a pool of particles. They are in a big volume. Some of them are in the inside volume. Some of them are outside. So the average of this count is the fraction of the volumes. If A equals to R, this will be 1. It makes sense. All the molecules will be inside the volume where they are confined. So it makes sense. All right. So Wj average is a cube divided by r cube. So we know this. And this, when n is large, so this is another approximation, n is large. This is n minus 1 is like n. So it's n squared a cube divided by r cube. So this is actually to the power 2. So it's average to the power 2. So this is to the power 2. So this is n a cube divided by r cube to the power 2. With this, we calculate the first term of the correlation function. All right, and I'm not sure if I will be able to calculate the second one. It's a bit more complex, but at least I will give you some hints. And I think in the next lecture I finish with that part. So the second part, remember, G2 of tau is, okay, the definition was the following. I was summing I equals 1 to n, and I come back, of Wi, okay? Wi, t, Wi, t plus tau. Okay, and then I'm doing an average. So you have to think about this. So in reality, here, what we are counting, this will be, many times will be 0, because a particle, this is the same particle that was at time t in the volume in the sphere SA. If it's outside at a later time, this will be 0, because it will be 1 times 0 equals 0. If it was out of the sphere, and then it, okay, so this one will contribute 0. If it was outside and entered, it will also contribute 0, because so the first case is Wit equals to 1, but it escaped, t plus tau equals to 0. This doesn't contribute to this sum. The second one was Wit equals to 0, so it was outside, but it entered, equals to 1. This doesn't contribute. So the only thing that will count is when Wit equals to 1 and also Wit plus tau equals to 1. This is the only contribution to this sum. It is when the particle was here, then it diffused it with whatever, but later, at a later time, it's also inside. Okay? So this is the main point. The way we can calculate this is surprisingly simple, at least the first step, then you will see it's not so simple. It is as follows. We can say something like this. This is a cube divided by r cube times u tau. And I will now explain what is u tau. So the first thing, but actually I can really unfold and do it in a different way. So we need to know these events. So we need, this is like a probability, because I'm just summing numbers that are 0 or 1. So I need the probability that a molecule is in SA at time t. Okay, this is not very good because in the sphere, in SA at t. Okay, so first you need this one. The molecule is inside at time t, and then the probability that it remains. So it is in SA at t plus tau, given that it was in SA at time t. Okay? So this one, it's quite simple because it is precisely this one. What is the probability among all the pool to find a molecule inside the volume? Is this one. It's a number smaller or equal than 1, this. So the first one, this one, is A cube divided by R cube. Okay? And the second one, I call it U tau. U tau will be, this one is exactly the probability that a molecule is in SA at time t. A molecule that, okay, that was in SA at time t is also in SA at time T plus tau. Okay, this is this quantity here. So if we can compute that, what is the probability that the molecule that was inside here later it's also inside here? So I'm taking two snapshots. Okay, this is the sphere A, and I found the particle here, and this is the same sphere. This was at time t, this is at time t plus tau. I still find the molecule here. Okay, this is a problem of diffusion. And this is what I will try to compute on the next day. Okay, so I don't know if maybe I can stop here, but let me just try to advance a little bit the calculation, and I'll stop in 5 minutes. So just to remind you, I had g tau. This is the total correlation function was g1 plus g2, so it was g1 tau plus g2 tau. I'm just wrapping up what we derived, and g1 was just m to the power 2, and g2 was m, the average, times u tau. Okay, when I wrote it with w's, but it's the same thing. Okay, it is the average times u tau, and in the other case, the first one, which was g1, is where is it? Yeah, m to the power 2. This is what I'm showing here. Okay, so m squared, and this is m times u tau. So now this means that, okay, now I'm going back to our goal. Our goal was to measure this one. Okay, I'm calculating this part, but what I want to do is the time integration. So I want to have the integration of this correlation function over these times. So now I calculated this, or at least I show you what is the expression. So it is like this. So now what I want to know is mt squared. This is the goal we want to do. It's calculating the second moment. Is 1 over t squared integral 0 t ds integral 0 t ds prime. And now we have this correlation function, which is just m squared, m average squared plus m average times u. And remember, if you trace back, here we had u evaluated at s minus s prime. So I'm explaining now how to calculate the correlation function for any tau. Then you just have to replace the tau by s minus s prime. This is what you need to do. Okay. The nice thing is this is a constant. This doesn't depend on s prime, so you can take it out of the integral. So you can write all this as mainly mt squared. Actually, this is mt. It is mt squared. Then there is the, okay, so the first term I'm saying is this is a number that you take out of the integral, and you integrate s from 0 to t, s prime 0 to t. So there is t times t squared, which goes with this t squared. That's why from the first term you get this. From the second term, you get the m goes out, and you get 1 over t squared integral 0 t ds integral 0 t ds prime u s minus s prime. Okay, so this is what we arrived. In the end, it is important to see that what we need to calculate is this u. So this is, I put it here in green, just to let you know what we will do in the next lecture. So I told you we need to calculate this, but we didn't calculate it. So for the next lecture, we will calculate this quantity, which is the probability that a particle which was in a sphere after diffusing around, it is yet inside the sphere in the next snapshot. Okay, next lecture. So once we know this one, we will be able to calculate mt squared, because this one we know, the average we know, this average we know, and we just need to do this. So I think this is a good time to stop, because otherwise it will become very formal and complicated. But please, before the next lecture, go back to the notes. And I think this one is, okay, it's not so trivial. I'm checking. It is related to an electrostatics problem. So you can also go through through the paper and look, but it is not so simple. But just I want to keep it here and in such a way that in the next class, I will start with this problem, which is calculating the probability for a particle to be in a sphere at time t and also to be in the sphere in a later time. Okay. Okay. So, any question, doubt? I'm recording, so things go well. No questions? All clear? Okay, maybe it's all unclear, but it's good you recheck everything and see that you follow my calculation. Please go to the reference I gave you, this paper, okay, which is what I'm discussing today. And please check if you have big doubts, you can go to this reference and look for it, okay? Actually, what I'm going to explain next day, it is in the appendix. It's not in the main part of the article, but calculating this is in the appendix of the paper. It's at the end, okay? All right, so if there are no more questions, I will, by now, stop the recording.